Introduction to Diagnostic Mycology. So mycology is the study of fungi, and fungi is a diverse group of organisms that's classified as molds and yeasts. Categories: classic pathogens and environmental saprobes, living on non-living material. May cause a range of signs and symptoms, mild to life-threatening, significant in the immunocompromised patient. So normally, a lot of fungal infections don't cause any problems in fully healthy people. There's just like a handful of true pathogens, but a lot of these are going to be opportunistic. But because in the hospital we see a lot of immunocompromised or sick patients, we need to be aware of this and be able to identify the different molds that are infecting our patients and the different yeasts that infect as well. So fungi are eukaryotic, which means that they have a nucleus. They have a nuclear membrane, they have all the organelles, they have mitochondria. They don't have chloroplasts though, so they're different from plants in that they don't have chloroplasts. They must absorb nutrients from the environment. They have thick cell walls made of chitin. So chitin is the material in their cell walls, where in bacteria we would typically be staining for the peptidoglycan. When we look at fungi, it's chitin. So I've repeated that multiple times because chitin is really important to know that that's in fungal cell walls. Preferred neutral pH. Moisture is necessary for growth, but spores and conidia survive in dry conditions for extended lengths of time. Yeasts are single vegetative cells. They form a smooth, creamy, bacterial-like colony without aerial hyphae. Identification is primarily based on biochemical testing and molecular diagnostic methods. Reproduction via budding or fission. So here you can see a yeast cell, and then it grows, it replicates its genetic material, and it buds off this like baby yeast. And eventually the baby yeast will grow and grow and grow larger until it buds off completely. Fission would be it just grows really big and gets cut in half. And that's kind of how bacteria divide. Molds, on the other hand, are fuzzy or woolly in appearance because of the formation of mycelia. Mycelia support the reproductive structures that produce conidia, and conidia are used to identify different fungal genera. Aerial mycelia extend above the surface of the colony, and that's responsible for the fuzzy appearance. Vegetative mycelia extend downward into the medium to absorb nutrients. Sometimes that can cause a mold colony to actually embed itself down deep into the agar, also known as pitting into the agar, so it'll actually sink down below the surface of the agar. Microscopic appearance often aids in the identification of molds. So yeasts are your single-celled forms, molds are typically your multicellular forms. So this is a visual representation of aerial and vegetative mycelia. So the vegetative mycelia are growing within the media, and the aerial mycelia are kind of extending up into the atmosphere here. And you can see that those aerial mycelia make it kind of look like it's a fuzzy appearance. So more about mold morphology. It's important to be able to distinguish the different features within the mold, because that's how we diagnose most mold infections. So we look at the spores and we look at the hyphae, and then we identify it based on structures that are common to particular molds. There are some other procedures, but they're a little bit more complicated. So if we can grow it up on a plate, we look at it under the microscope and we can very easily distinguish the different species. So hyphae, we look at the shape, whether it's septate, sparsely septate, or aseptate. The shape would look like an antler, a racket, or a raquette, spiral, rhizoids. Septate, frequent perpendicular cross walls. Sparsely septate, frequent perpendicular, or infrequent, that should say, infrequent perpendicular walls. This is what happens when you use the slides provided by the publisher. Sorry about that. I just want to make sure it's accurate when we go forward. So this is septate. So you can see the cross walls in here. And this is sparsely septate. So not a lot of cross walls. You may encounter one here and there, but for the most part, there's not very many. And aseptate, there's none. So here you can see septate has the cross walls in the hyphal strands, and then aseptate, you don't see those. Hyaline versus phaoid. So hyaline is non-pigmented or lightly pigmented. Phaoid, also known as dematiaceous, and this is how I learned it, is dematiaceous, is darkly pigmented, and that's due to melanin production in the cell wall. That dark pigmentation can help prevent UV damage in the cell, so it's important to kind of have that pigmentation, especially if it's going to be a fungus that grows out in the environment. Dimorphism and polymorphism. So dimorphism is the ability to exist in two forms based on the growth conditions. So the mold phase would grow if you incubated at around room temperature in ambient air. The yeast phase would grow if you incubate it in body temperature with increased carbon dioxide. So if you simulate the environment of the human body, it's going to grow in the yeast phase, while if you simulate the environment, like the environmental conditions, room temperature, your typical atmosphere, it's going to grow in the mold phase. And there's certain pathogenic organisms that are dimorphic. So depending on how you incubate them, they're going to grow in a different way. Polymorphism is the ability to form yeast and mold within the same culture. Generally, yeast is first produced and then it eventually turns to mold as the culture gets old. Exophiala species are one of those species that does this. Reproduction. So as I mentioned, one of the main ways to identify molds is not only by the hyphae, but then by the spores that are produced and the reproductive structures that produce those spores. So asexual reproduction is predominant for fungi. Asexual means that it doesn't need to actually combine its genetic material with another organism of the same species in order to create offspring. So it can just reproduce without actually recombining its genetic material with another. This is how bacteria reproduce. So we don't have bacteria coming together, forming bonds and then making babies together. No, bacterial cell just grows really big and splits in half. For the most part, that's going to be how molds reproduce and fungi in general reproduce. There are some instances where sexual reproduction occurs, and we'll talk about that briefly. But we have conidia, or the conidium, following mitosis. Conidia are fruiting structures. They're conidiogenous cells and annelid ring structures. Then we have phyalides or phyalidase, vase-like structures that produce phyaloconidia. Annelides are ring structures that produce annelidoconidia. This is a diagram that shows you what these different parts are. So in a typical hyphae, this, you can tell it's septate hyphae because it has these cross bridges here. It would randomly grow up these structures on a conidiophore, and then it can form this phyalide, and the phyalide can burst open and release conidia, and conidia are spores. These are like perfectly packaged genetic material deposits. So when these land in a fresh new area, they can grow into a whole new mold organism. It's going to be genetically identical to the original because they're reproduced through asexual reproduction. Another type of conidia is the arthroconidia, singular arthroconidium. These conidia are formed by the fragmentation of fertile hyphae, as opposed to being formed by conidiogenous cells. In the clinical laboratory, mold identifications may be based on the structures formed as a result of asexual reproduction. So you would take a hyphae, and this isn't typical hyphae, this would be, as it mentioned, fertile hyphae. So the hyphae would kind of chunk off, and then all of those chunked off hyphae would be arthroconidia. You're going to see some examples of these as we go through the rest of the Mycology series. Sexual reproduction requires the joining of two compatible nuclei followed by meiosis. There's a telomorph, a fungus that reproduces sexually. Occasionally, these fungi will reproduce asexually as well. The asexual form is called an anamorph. Synomorphs are named for amorphic strains when more than one amorph is present for the same telomorph. So the zygospore formation is kind of an example of this. So here you have hyphae 1, hyphae 2, where the hyphae from the two different organisms meet, they fuse together, and then the nuclei fuse, and then they produce a zygospore. And then the zygospore will undergo meiosis, producing a haploid organism after that. Taxonomy. So the causative agents of clinical infections are found in 4 groups of fungi: the phylum Ascomycota, the phylum Basidiomycota, the subphylum Mycotina, and form division fungi imperfecti, and that's Deuteromycota. For the Ascomycota, approximately 50% of all named fungi are classified in this phylum. The sexual spores, ascospores, that are formed in a sac-like structure known as an ascus. Representative species would be Microsporum, Trichophyton, and Cetiosporum. We will be looking at those, all 3 of those, in detail as we go through this mycology series. Basidiomycota. Major pathogens are members of the Cryptococcus neoformans and C. gaddii species complexes. Cryptococcus neoformans is a yeast, and we're going to talk about the significant infections that guy causes. Members of these genera are associated with human infections: Malassezia, Trichosporin, and Rhodotorula. Basidiomycetuous molds are being recovered in increasing numbers in the laboratory, but their clinical significance is not clearly understood. Close communication between the physician and the laboratory would help determine whether the isolate is an environmental contaminant or an agent of disease. Mucorales, the most significant order. These are rapid growers, rapidly growing organisms normally found in soil. They produce gray to white aerial mycelium with hyaline, sparsely septate hyphae. They are opportunistic pathogen in the immunocompromised. Clinical infection is referred to as mucormycosis. Reproduction, asexual reproduction, sporangiospores, asexual spores are produced in a structure known as a sporangium, which develops from a supporting structure known as a sporangiophore. Although some mucorales are capable of sexual reproduction, resulting in the production of zygospores, these structures are not routinely seen in the clinical laboratories. Examples of clinically significant genera: Actinomucor, Mucor, and Rhizopus. So here is asexual reproduction by Mucorales. So here you can see there's a sporangiophore that's formed. At the top of a sporangiophore is the sporangium and the columella. The columella is surrounded by the sporangium, and then sporangiospores will be produced inside of here, and eventually this breaks open and releases those into the environment. Fungi imperfecti. So it contains the largest number of causative agents of mycoses, including cutaneous, subcutaneous, and systemic diseases. Organisms are placed in this group when no mode of sexual reproduction has been identified. These organisms are identified based on the basis of characteristics asexual reproductive structures. Mycoses. So mycoses are the diseases caused by fungi. Disease categories are superficial, cutaneous, subcutaneous, and systemic. Those are bolded because those are important. Systemic are infections not involving the skin or deeper tissue, just under the skin. So systemic, I kind of think of those as being body-wide. They can impact multiple organ systems and different areas deep within the body. Superficial and cutaneous mycoses are caused by fungi that can degrade keratin, dermatophytes, and fungi that cannot degrade keratin, non-dermatophytes. Dermatophytes and non-dermatophytes are an important subcategory, so you need to know the difference and be able to define those. Superficial would predominantly be on the surface of the hair, skin, and nails. Cutaneous can be within the hair, skin, and nails. So this is a diagram of skin tissue in layers which fungal infections can occur. So here's the keratinized layer at the top, so your superficial ones would be there. You can have the epidermis here, your your cutaneous ones would be there, and then deeper into the subcutaneous layer, which is under the bottom layer of the skin, and that would be the subcutaneous infections. Superficial mycoses are infections confined to the outermost layer of the skin. Because infections of the skin, hair, and nails were at one time believed to be the result of burrowing worms that formed ring-shaped patterns in the skin, the term tinea, in Latin meaning worm, was applied to each disease, along with the Latin term for the body site. Common terminology, ringworm. Tinea dermatophytosis is ringworm. Tinea versicolor is pityriasis versicolor, discoloration or depigmentation and scaling of the skin. It's caused by Malassezia furfur. Sounds really fun to say. They call that the spaghetti and meatballs type of fungi. Apparently, to some people, it looks like spaghetti and meatballs under a microscope. We'll talk more about that one later. Tinea nigra, non-dermatophytic, almost always caused by Horte Wernickeii, and is characterized by brown or black macular patches, primarily on the palms. Biopsy and culture of the skin are important to distinguish this infection from melanoma and a much more serious non-fungal disease. Melanoma is, of course, if you know, skin cancer. Another superficial infection, Piedra, is confined to the hair shaft and is characterized by nodules composed of hyphae and cement-like substances that attaches it to the hair shaft. Black Piedra is caused by Piedra horte. White Piedra is caused by Trichosporon ovoides and T. incen. Onychomycosis caused by Onchocola canadensis and O. canii is also a non-dermatophytic superficial mycosis characterized by white superficial and distal lateral sublingual infections, or subungual infections of the great toe. We will be going through black and white Piedra and when we talk about those superficial mycoses. Cutaneous mycoses. So dermatocoses are defined as fungal diseases of the keratinized tissues of humans and other animals. Although non-dermatophyte species are capable of causing similar infections, this syndrome is usually a result of infection with a dermatophyte, hence the term dermatophytosis. This term should not be used until the causative agent has been identified, though. Dermatophytic infections usually involve a restricted region of the host. Traditionally, these diseases are named with respect to the portion of body that is affected. So, tinea capitis refers to the head. So that would be like the ringworm of the head or a fungal infection of the head. Tinea favosa, head, but that's a distinctive disease. So it's different from tinea capitis. Tinea capitis is literally dandruff. So if you've ever had dandruff, that's what that is. Tinea barbae, confined to the beard. Tinea corporis, the body, so the glabrous skin. Tinea manum, or manuum, is the hand. Tinea unguium is the nails. Tinea cruris is groin, also known as jock itch. Tinea pedis is in the feet, also known as athlete's feet. Tinea imbricate, body, in a distinctive lesion. You do need to know these locations of the body and the tinea that is associated with them. Each ringworm lesion is the result of local inoculation of skin with the causative agent. Lesions enlarge with time, usually with the most inflammation occurring at the advancing edge of the lesion. Some cases of ringworm are subclinical, exhibiting only a dry, scaly lesion without inflammation. Symptoms of cutaneous mycoses would be itching, scaling, ring-like patches of skin, thick, discolored nails, brittle or broken hairs. Example members would be trichophyton, epidermophyton, and microsporum. Subcutaneous involve the deeper skin layers, muscle, connective tissue, bone. Generally, dissemination through the blood does not occur. Clinical feature, progressive non-healing ulcers, presence of draining sinus tracts, varicosus nodules that often become, or verrucous nodules that also become ulcerated and crusted. Chromoblastomycosis and eumycotic mycetoma are different types of subcutaneous mycoses, and we will discuss these more once we get to that region of our lecture series. Sporotrichosis, caused by Sporothrix schenkii species, commonly presents as progressive lymphocutaneous infection. It begins with a single draining lesion and progresses along the limbs via the lymphatic system. It forms multiple draining lesions. Granules are not formed, thus not qualifying this infection as a mycetoma, but its dissemination of these species causing systemic sporotrichosis is much more common and has been implicated in pneumonia that is refractory to antifungal therapy. If it's refractory to antifungal therapy, that means it has the potential to avoid treatment by antifungal therapy. So we don't necessarily call it resistant, although there are forms of resistant fungi out there. We call it refractory instead of resistant because the fungi themselves don't actually have the typical resistance mechanisms that you would find in like a bacterial cell. They are refractory because the drug just doesn't work on them in general. Systemic mycoses, infections that affect internal organs or deep tissues of the body, frequently enter the lungs and then spread. Generalized symptoms: fever, fatigue, sometimes chronic cough and chest pain. Historically, the term systemic mycoses was used to describe diseases caused by thermally dimorphic fungi, histoplasma, coccidioides, blastomycetes. Those are really bad guys that we're going to talk about in this course. However, other fungal agents are also capable of causing systemic disease, including but not limited to Aspergillus, Fusarium, Cedosporium, and Curvulera, as well as some yeasts such as Candida and Cryptococcus. All of those that were just listed are species we're going to talk about in this course. It is important to note that any fungus is capable of disseminating from the primary site of infection in the immunocompromised host. So here is a classification scheme that you can take a look at. You can pause the video and look at the different species that are represented. So basic mycology techniques. First, safety procedures. Molds produce spores, and spores go everywhere. They can be inhaled, they can land on someone's skin, they can go all over hospital environments. And when we have sick people coming into the hospital for treatment, we do not want them to get an opportunistic mold infection. So we use extra careful precautions when using anything that has to do with culturing of molds. So similar requirements as working with mycobacteria. We have that negative pressure room. There's a double door system typically. And the whole goal of that is to prevent the spread of those spores. There is a designated space. So it's like the mold culture room, and nobody enters in there except for the tech that's designated to working with mycology. Mold spores, again, are the primary concern, and they require a lengthy incubation time. You must seal the plates to increase safety and prevent contamination and/or exposure. Also, to prevent them from drying out, because we incubate them for so long, we don't want them to dry out. Typical specimens at the mycology bench. So specimen collection, handling, and transport, antimicrobial containing media to inhibit bacterial growth. If a doctor orders a mold culture, we don't want bacteria in there. Historically, bacteria and molds have fought each other. That's where antibiotics come from. So it was a mold called penicillium happened to land on a plate of bacteria, and the penicillium mold produced a toxic compound that killed off the bacteria. So anywhere the bacteria was growing next to the mold, it died. And we're like, wait a minute, if we could isolate that compound, we could actually use it in human medicine. Ancient Egyptians used to rub moldy foods on wounds to try to heal themselves because they knew that there was some type of healing power in those. Penicillium had that antimicrobial activity to it. So if we have bacteria and molds growing together, bacteria are going to do the opposite thing to molds. Bacteria are going to kill off the molds. So there's like this constant war between molds and bacteria because typically they want to eat the same thing. They want to decompose dead things. And If we have bacteria present, it's a contaminant and we don't want it around. We just want to culture the fungus. So a lot of times we'll add antimicrobials into the media. Specimens that we would commonly get would be hair, scrapings, so skin scrapings, nails, for the KOH prep. Blood and bone marrow lysis centrifugation system releases the organisms from the cells. Cerebral spinal fluid, 1 drop for India ink or latex test for Cryptococcus neoformans, plated directly because CSF should be sterile. Abscess, wound, or tissue, mince and grind the tissue, plate directly or perform other tests. Respiratory specimens, plated directly or digested with mucolytic prior to plating. Urogenital or fecal specimens, culture if urine first voided morning urine, centrifuge to sediment. So here's some common pathogens and the body locations where you might recover them. And here is the kind of breakdown scheme of how we would go about diagnosing an infection. So once we get a clinical specimen in, we can decide, are we going to do direct examination or culture? And maybe we'll do both. So we're going to talk about the processes of what's involved with direct examination, and then we're going to talk a bit more about how you would culture them and what you would look at after you culture them. So culture procedures, there's specialized culture media that we use, sabro-dextrose agar or SDA, with or without antimicrobials. Potato flake agar, PFA, brain heart infusion agar, BHI, enriched with blood and antimicrobials. Plates or tube media, tubes are safer to handle and less susceptible to drying. A lot of times those are preferred. If we need to see colony morphology though, it's really tough to see that in a tube. So then we would have to culture it out on a plate. Incubation would be room temperature or 30 degrees Celsius. I've seen 30 degrees Celsius more often in clinical labs than 25. And dimorphic species, or if we suspect dimorphic species, we would incubate a plate at 37 degrees Celsius as well, and then maintain for 4 to 6 weeks. There are some rapid growers. If you remember going through the slides here, some of them are rapid growers, and those will produce results a lot faster. So these plates need to be checked often, and there's protocols for each type of culture based on its source that a mycologist would follow. Other types of agar, birdseed agar, it's selective and differential isolation of Cryptococcus neoformans from other yeasts, including other Cryptococcus species. Sabro-dextrose agar, I already talked about, that's just a general fungal growth medium. Potato dextrose agar, PDA, more for food testing than clinical testing. Chrome agar is specific for yeasts, so it's going to turn different colors in the presence of different yeasts. Dermatophyte test medium, or DTM, contains cyclohexamide to inhibit saprophytic growth. That means the growth of our common environmental molds. So we don't want the environmental stuff to grow, we only want the ones that grow in the skin to grow. Antibiotic to inhibit bacterial growth, and a phenol red indicator, which is a pH indicator. So these are various species growing on different types of agar. Cornmeal agar is another one. It allows us to look at the chlamydospore production from Canada albicans, or Canada species in general. Culture procedures. So this is the slide culture method. Sometimes we don't want to inoculate it on an entire plate. We don't want to inoculate it on a slant or in a tube. We want to just look at a small growth of one colony of the organism. We can create a slide colony or a slide culture for that purpose. So if you want to go ahead and make a slide culture, this is what you would set up. So there would be a slide, you would put a little chunk of agar on the slide. Typically you would moisten the bottom, you would put water in the bottom, or you would put a wet paper towel on the bottom so that there's some moisture and humidity inside of here. And you would put this inside of a petri dish and then put these two I would just break apart a cotton swab. Unless it's water, then you'd have to use some type of plastic stick. I'm sure they make specialized structures for this, but when I performed it last, which was quite a few years ago, I just broke the cotton tip off of the cotton swab, and that's what I used to elevate it. And the real purpose of elevating it is because we want the bottom to stay moist, but we don't want the slide to get wet. So what you would do is you would take a small amount of the mold and you would stab it into the agar, and then you would put a cover slip on top of it, and then you would put the cover on top of the petri dish and incubate it. After you see some growth, you would then come back and lift up the cover slip carefully using tweezers, and then you'd put a drop of LCB stain, lactophenolcotton blue stain, on the slide, and then you would drop the cover slip that has the mold spores, because the mold grew literally up to and under the cover slip. You would drop it directly on into that LCB stain, and then you'd look at it under the microscope. You can make these permanent smears if you just put some nail polish around the edges. So then that would be a permanent fixed smear. You let it dry, and then you can look at it for years to come. Lactophenolcotton blue. I am going to talk a little bit more about that now. I may mention it later in the slides, can't remember, but this is an important stain that I want you to know about. The reason why we use lactophenolcotton blue is because it kills spores. So spores are really dangerous. We have to be careful when dealing with spores. And in this class, all of our mold manipulation is going to be performed under a hood. So when we work with spores, we want to be able to kill them quickly. We still want to be able to see their structure though. So unlike bacteria, where we can just like heat fix them to a slide and that pretty much kills them, if we light spores on fire, they're going to be disintegrated. They're going to be destroyed completely. So what we do instead is we use this LCB stain. Lactophenolcotton blue kills the spores and makes this slide safe to remove from the hood. So now you can remove it out of the hood and look at it under a desktop microscope. So macroscopic morphology. When we look at the colony, we have to report several different pieces of information. First, the colony growth time. We definitely want to report when this thing actually started producing a visible colony. The color, the texture, the pigment on the reverse side of the colony. So we look at the top color of it and the color that it looks like from the bottom of the plate. The texture could be found on the surface, so like smooth and glistening, rough, wrinkled, dry, powdery. Clumpy would be like flocose. And then the elevation, including whether or not it is submerged into the agar. You want to do the whole colony, shape, the edge, shape, elevation, and surface. So lots of information that we have to report. We will be doing this we will be looking at colony morphology when we look at the molds. KOH preparation. So potassium hydroxide preparation, 10% to 20% solution of KOH. Some may contain dimethyl sulfoxide, DMSO, and a stain. Equal parts sample to KOH. Cover slipped and heated gently. The purpose is to dissolve away the keratin and visualize dermatophytes. So potassium hydroxide, 10 to 20%, will completely dissolve away your skin cells and any bacteria that's there. But it leaves the fungal elements because the fungal elements have chitin in them. It doesn't destroy those. So we can see the fungal elements that are left behind. Sometimes we add in chalcofluor white. Chalcofluor white is a dye that binds to cellulose or polysaccharides present in chitin. Apple green or blue-white in UV light. So here you can see the chalcofluor white stain and the fungal elements present. India ink. India ink is a negative stain that will stain the background, and that allows you to visualize the capsules of yeast or bacteria. This is a picture of an encapsulated yeast, Cryptococcus neoformans. Tissue stains, periodic acid Schiff, PAS, GMS, hematoxylin and eosin, Giamza, or Fontana Mason stains. So these are the colors of the fungal element and the background color. This is really good for reference purposes. You don't necessarily have to memorize all of the different colors. So microscopic evaluation, then you'd look for septate versus sparsely septate versus aseptate, hyaline or dematiaceous hyphae, fruiting structures, the type, size, shape, and arrangement of the conidia. A tease mount. Sometimes you can do those slide culture preps, and you can just pull up the cover slip and plop that down on a slide of LCB and look at it under the microscope. Other times, we want to just directly look at the mold that's growing on the surface. But if you've ever actually manipulated or handled molds, molds are really difficult to pull apart. That structure grows so tightly together that it's really hard to get apart. Also, a lot of molds are waterproof. So if you get a big mold colony, they're waterproof because they're super bound together. So one of the procedures we can do is a tease mount. Tease mount is exactly what it sounds like. We take a toothpick and try to tease it apart. We remove part of the colony, we mount it in a drop of lactophenylalanine or cotton blue stain, and then tease apart the colony using a teasing needle and overlay with a cover slip to disperse it. Cellophane tape preparation. So we would take clear tape, press it on the surface of a colony gently but firmly, and then we would put it on top of a slide with a drop of lactophenol cotton blue. We stretch the tape across the slide, lowering it into the stain, pull taut, and affix the other side of the tape, avoiding air bubbles. So when we do that, the tape serves as the cover slip for the procedure. So here you can see the tape that's wound around the finger of this tech. Then they would put that tape directly on top of the LCB stain on the slide, and they would just kind of move apart the tape to fix it down flat to the slide, and then they're going to look at it under the microscope, and the tape serves as the cover slip. Miscellaneous tests for molds, the hair perforation test. So here's a picture of that hair perforation test. Basically, you would have to take really thin blonde hair, which they get from a variety of sources, and you would take your unknown pathogen and culture it on top of this hair. And certain pathogens have the ability to cause the hair to perforate like this. So if the pathogen grows on the hair and it causes these perforations, we can visualize that after we stain the hair. It comes up pretty obviously, and then we can diagnose the species that way. The urease test, just like we do for bacteria, this is the urease test. A hot pink color would be positive. We can test for a thiamine requirement, trichophyton agars, growth on rice grains, and other miscellaneous tests. Miscellaneous tests for yeasts, germ tube production. So Candida albicans produces a germ tube. So here's your yeast cell and a germ tube. If exposed to serum, this yeast cell will grow out this germ tube, and it looks like a tail that grows out of the yeast cell. So if that happens, then we kind of know that it's Candida albicans. Carbohydrate assimilation, chromogenic substrates, and cornmeal agar colony morphology. We can also throw yeasts on the MALDI, and the MALDI will identify as well. There's also yeast-specific panels now for the microscan. So here is the yeast differentiation, and this growth chart will, schematic diagram will be good when it comes time to learning a little bit more about the yeast. Other laboratory methods, immunodiagnosis of fungal infections, anti-sera to the cell wall antigens, antifungal susceptibility, so antifungal agents. The main agent is amphotericin B. Many side effects, fever, rigors, renal impairment, and possibly shock. There aren't a lot of antifungal substances. In order to produce an antifungal or an anti-anything, we have to have something to target. So like antibacterials are really easy to, or antibiotics, which target bacteria, really easy to create because we can target peptidoglycan, because human cells don't have that. We can target the ribosome because human cells' ribosomes are different. So there's structures in bacteria that we can target because bacteria are so different from us. Well, our cells are very similar to fungal cells because our cells are eukaryotic organisms, and so are fungal cells. So it's difficult to find something to target. Sometimes we can target chitin, but that only goes so far. So we need other types of targets, and it's really difficult to find those because of the similarity between yeast cells and our cells. It's easy to develop topical antifungals that only just go on the surface of the skin, but once you start to try to consume a pill, that makes it tough because now that thing has to go through your body. Other agents include fluconazole, itraconazole, posaconazole, and voriconazole. You can see azole at the end of that typically indicates that that is going to be an antifungal agent. So now I'm going to go briefly through the Aspergillus, Penicillium, and Rhizopum, or Rhizopus. These are the three kind of go-to mold species that you would commonly find out in nature. Some of these do have the ability to cause disease. I'm not going to talk about the diseases that they cause because this week's lab is going to focus on the basic mycology lab procedures. So I'm going to show you some structures to look for to be able to identify them, but we're not going to go through necessarily the diseases that they cause. So Aspergillus, there's 4 different types of Aspergillus, or 4 main types that we worry about, and you can see those 4 main types that are pictured here. The colonies look different, and so do the reproductive structures. Aspergillus has this conidiophore. You can see the septate hyphae, and that's going to grow up into this huge structure. You have Phyalides, which will produce conidial chains, and these chains kind of go up. And then eventually these individual cells and these spores will slough off. So you can see the chains, so you have the Phyalides down here, and then you can see that the chains are growing up, and then these are loose spores that have been released. A. fumigatus, A. niger, and A. flavus have very different types of structures, but they're easily recognizable under the microscope. Penicillium. Penicillium looks like a tulip to me. Penicillium is distinctive from Aspergillus because Aspergillus has a really wide head. You can even see on these pictures, like the head is pretty wide and thick and full looking. Penicillium has like a countable number of spores that are coming up here. So even though they look similar, you can kind of start to see the difference. This is less full looking and countable numbers of these spores on the top. So Penicillium, different species are going to come in a wide variety of different colors, but in general, their structures, their reproductive structures are going to look like this. So we have Phyalides, and then those are going to go up to spores at the top. This is what Rhizopus looks like. This is probably, so this powdery surface or whatever, this is probably the most commonly encountered mold in the environment. This is going to be all over basements and stuff. It's everywhere, difficult to avoid. A lot of times when we see Rhizopus, it is just a contaminant. But when you look at it under the microscope, Rhizopus has this very distinctive circular structure at the top. So you're not going to see like branches coming out of it. You're just going to see a circle. And here you can see that again. So this under the microscope. The sporangula are columella and globose. Sporangiophores are at the nodes directly above the pigmented rhizoids. So here you can see Rhizopus. You've got this sporangiophore, the columella, and the sporangium releasing the sporangiospores. And that is it.